for joining today to hear about intracellular phase separation and magic numbers. It's part of the Physics of Life online summer school hosted by the Center for the Physics of Biological Function and the NSF. I'm Ned Wingreen. I'm a professor at Princeton in molecular biology. My background is in theoretical physics, and my group does theory and modeling of a biophysical nature for living systems. So let me first tell you what I'm going to tell you. So here's an outline for today's talk. I'll begin with an introduction to phase separation taking place both in cells and in vitro. That means in glass, so in the laboratory outside of cells. And then I want to focus on an area that's been of particular interest to me and my group, which is phase separation when there are two components. And I don't mean just a solvent like water and a polymer, but actually two polymers that actually have to get together in order to phase separate. And multivalent means that they have many interaction sites on each polymer. So each polymer will have many, many sticky sites on it to interact with other polymers. And of course, condensate is sort of another word for the dense phase in a phase separated system where, for example, lots of polymers get together. I'm going to focus on a couple of examples during the body of the talk. An in vitro system of engineered polymers, and I'll explain exactly what SIM and SUMO are. And then I want to make sure that we get to a very exciting in vivo living system called the algal Pyrenoid. Now, before working on this, I had never heard of a Pyrenoid. It turns out that about 30 to 40% of all of the CO2 that's fixed out of the atmosphere every year, turned into organic carbon for animals like us to eat, takes place in little organelles called the Pyrenoid. And it's a fascinating example of one of these phase-separated systems. And in the last moments, I'll draw some general conclusions so you have some take-home messages. So I wanted to start with a kind of broad introduction by giving a couple of examples of cases where phase separation is playing an important role in living systems. And I should point out these are complicated examples, and we'll try to answer any questions you might have about them, but I don't make any promises in particular since these are not actually systems that my group specifically works on. But let's dive in. These were both mentioned in that paper by Brangwyn and co-workers. So we'll dig in a little deeper to those systems. And the first one is something called C. elegans. And so this is a picture here in your upper left of a C. elegans, which is a nematode worm. It crawls around in the soil, depending where you are. If you go out and dig in the ground, you'll probably find some C. elegans or their relatives dining on bacteria. Now, this particular C. elegans isn't ready to dine on bacteria. It's very young. It's still a single cell. It will eventually divide into many cells that will become the worm. But I want you to notice something about this single cell, which is that it's filled with green droplets. Now, these are green because people have engineered particular proteins that are being expressed by this organism to be green and fluorescent. And that's how we see them, but otherwise this is behaving normally. And what you see down below is that as this first cell gets ready to divide, some things have happened, and you see that the green droplets are all on one side of the cell. And in fact, when this cell divides, it's going to be an asymmetric division. So the two daughter cells are going to have different jobs. They're going to give rise to different lineages inside of the worm. And in particular, the right side here is going to have all of these little droplets, and those are eventually going to end up in the germline, in the reproductive cells of the worm, at which point the contents of those droplets will become very important. They're filled with RNAs that will eventually be translated into proteins. Now, these droplets have been known for a long time, and they were given the name of P granules. And so when you think about what is a granule, the answer is that a granule isn't really a science word. It means you don't really know what it is. It's not like the phases of matter are solid, liquid, and granule. So it was kind of a mystery. What were these objects? And that remained a mystery until My colleague, Cliff Brangwyn, and Tony Hyman, who he was working with at the time, asked that question. Cliff came from a material science background, and he wasn't happy with the word granule. So he said, well, what are these things, as materials? And so by doing some very simple experiments later on in the life of the worms, for example, taking these germline cells and just putting them between glass slides and looking at them, he saw the following. And I'll show this movie more than once. So these light-colored regions are those key granules. And you see that when they're subject to shear forces by being squeezed between glass slides, they do stuff that looks just like liquids. It looks like there's a light-colored liquid mixed in with a dark-colored liquid. And you're seeing that liquid, let me show it one more time, you're seeing it flow, you're seeing it drip, you're actually seeing some cases where it merges together, droplets get together and merge. And so from experiments like this and other very nice, elegant, simple experiments, Cliff was able to make the case that these things, which we'd call granules, are actually liquid droplets. And so what's happened inside this living organism is some of the material, some of the protein, and in this case, some of the RNA, have gotten together and phase separated into liquid droplets, just in the same way that if you had a mixture of oil and water, the oil would condense into droplets inside of the water. And so let me show you as a schematic what's actually happening inside of this C. elegans cell, which is really a kind of beautiful little experiment happening inside this single cell. And the idea is that this cell has a polarity. It has two different ends. A is for anterior and P is for posterior. And these are established actually by the entry point of the sperm into the egg. And so a lot of things happen in the cell. Lots of different proteins live at the A end. Lots of different proteins live at the P end. And what these do is that they establish a gradient inside the cell of the material that goes into these droplets. And so the idea is very much like oil and water. So if I'm sitting at this end of the cell, it's as if I have a solution where I put just a tiny bit of oil into water. And when I do that, because of entropy, the oil will actually dissolve in the water and it will be just spread out and it'll be one homogeneous mixture of a little bit of oil and water. But if I keep increasing the amount of oil, which is sort of like moving this direction in the cell, then I will reach a certain concentration of oil and water, and actually any additional oil can't go dissolve into the water, and instead it will start forming droplets. And so what you see inside the cell is exactly that, where the concentration of these components is low, it's dissolved, where the concentration becomes higher, all of that extra material, as if that extra oil that I had put into the water, goes into a condensed phase of droplets. Now, the sizes of these droplets really depend on how long this system has been going on. Droplets will tend to grow and to combine with each other and become larger. And the reason we don't see one large droplet is because all of this is sort of happening in real time. These droplets have just started to form and will start to condense and only be partly coalesced into single droplets by the time this cell divides. And so the take home from this is that even though this is a very complicated system, there's thousands and thousands of different proteins and RNAs, we can understand this aspect of it in terms of very simple physics, no more complicated than oil and water. And as a result, we can see that the cell doesn't need to move these droplets around using motors. It just needs to establish a simple gradient of material, and that will be enough to concentrate all of these RNAs and their associated proteins on one side of the cell. So when the cell divides, they're all in a particular daughter cell. So I'm going to move on now from C. elegans to talk about another example, just to give a sense of the breadth of the importance of phase separation inside of living organisms. And so the next example comes from another model organism studied in many labs, which is the frog, Xenopus laevis. And so the Xenopus laevis, in addition to being an interesting organism in many ways in its own right, has some very useful properties. It lays tons and tons of eggs, and these are very large. So people are concentrating the eggs, breaking them down, are able to study what's happening in this living system in a kind of in vitro or semi-in vitro way by extracting them from the eggs and looking at what happens with very high concentrations of material so you can actually do biochemistry. Now, in this case, what's being done instead is imaging. And again, I borrowed this from Cliff Brangwyn, who's really one of the heroes and leaders of this field of intracellular phase separation. And what you're seeing here are non-membrane bound organelles. I think you heard about this from Jun Zhang if you joined us this morning. These are the nucleoli. They live in the nucleus of cells, and their job is to process the RNAs that get eventually turned into ribosomes. Ribosomes are the machines that translate messenger RNAs into proteins. And so in this early stage of the frog, when it's going to be developing into a frog, it needs lots and lots of ribosomes. So it needs lots and lots of these nucleoli in order to manufacture ribosomes. Now, what are you seeing? You're seeing again fluorescence because two different proteins that go into these nucleoli have been labeled. Some have been labeled with green fluorescence, some have been labeled with red fluorescence. And it looks a little bit complicated, like there's little greenish domains inside red domains. The reason it looks so complicated is because there's also a cytoskeleton. There's polymers that have formed filaments made of actin inside of these nucleoli. And what can be done is to add a drug that will actually make these filaments fall apart, leaving the molecules that phase separated. And you'll see what happens when those drugs are added. And you see that the green domains now start to form together, surrounded by these red domains. And so what you're actually seeing here is phase separation, now where there are actually three phases. There's the solvent phase, mostly water, and then there's this phase which is enriched in the protein labeled red and lots of other proteins, and another phase which includes the protein that's labeled green and lots of other phases. Lots of other proteins. And so even though this system is very complicated and has hundreds of different components, it's behaving, again, like a very simple system. You could imagine mixing three different substances, say oil and water and something else together, and they could form this kind of structure. And whether the green ends up inside the red or as separate droplets depends on the interactions between the green components and the red components, whether it's lower free energy to form components like this or to form separate ones. And you may have heard something about this earlier in the seminar series. So let me just show you, these are sort of lower resolution images. When you see higher resolution images, you really see these beautiful liquid phase separated bodies that really come right out of living organisms. So it looks like something you would have made in maybe a high school chemistry lab, but this is happening inside of living cells. So while it's wonderful to look at these living systems, try to understand how they work and why they work, it's also possible to reconstruct similar systems like this using man-made components, or at least partially man-made components. So I'm going to show you an image here taken by my student, Daniel Lee, who works with me and Cliff Brangwyn, using a system called the optodroplet system, where particular proteins are expressed by these living cells, and these proteins are at least partially designed by people to have certain properties. And the property they have is that when blue light is shined on these cells, certain proteins join together, and that's enough to cause the system to go from a regime where the components are not phase separated. When they join together, they form components that are now in a regime that phase separates. And so you'll see if I play this movie, the blue light comes on, and then all of these proteins that are labeled red will realize that now they're sticky enough, they want to phase separate. And so you see them start to coalesce first into little droplets, the bigger droplets, the little droplets join together. And so we can then study the processes and mechanisms and implications of phase separation in cells using these engineered systems. Now you may say, well, okay, that's great for experimentalists, but what about theorists like you and my group? Well, very challenging to study systems that compose of hundreds of components, or even the relatively small number of components that went into these optodroplets. And so this presents a challenge. But let me take a moment now to answer any questions. So I'm going to take a look at the questions that might have come in. So one question from Maya is, if cell division is blocked, could you see one big droplet forming instead of individual droplets? And the answer is basically yes. Your intuition is absolutely right. If I have two droplets, I can lower the free energy of the system by bringing them together to form one droplet, because that one droplet has less surface than those two droplets, and that lowers the overall free energy of the system. And we see this in those optodroplet case. The longer you wait, the more the little droplets either merge with the big droplets or dissolve in favor of the big droplets. So that's very much correct. Someone has asked, what is free energy and Gibbs free energy? Free energy, as is sort of the physicist's favorite quantity. It's a measure of the overall combination of entropy and energy in a system, and the world, when it reaches equilibrium, reaches a state of minimum free energy. Gibbs free energy is just a specific case where both the temperature of the system and the pressure of the system are considered fixed. And then one asks, well, given that fixed temperature and fixed pressure, then what is the lowest Gibbs free energy state of the system that it will tend to, as if I leave the system alone long enough to reach equilibrium? Yes, someone has asked, are there other factors except blue light that might cause condensation? The blue light is very specific to this engineered system. In other systems, yes, phase separation is definitely regulated. You'll have polymers that, for example, normally would not want to stick together, but the cell will modify them, add chemical groups to them, change their charge, and so on, that will then make them have more favorable interactions, and that will cause them to condense. And this is generally reversible. The cell can then undo those modifications so that phase separation now is no longer favorable, and these will come apart. And in fact, that's actually one of the themes in intracellular phase separation, is that these condensates tend to form and break up very dynamically in response to the needs of the cell. Let's see. Why do the proteins behave differently in the presence of blue light? In this particular case, there is one of the proteins, when you shine blue light on it, it enters an excited state, which changes its conformation, which then lets it interact with a different protein. And what kind of mechanism allows for the optodroplet proteins to forget their current phase and coalesce into droplets? Again, it's the mechanism where one protein opens up, it joins another, and actually this protein is often in the optodroplet system is part of a core. So there's 24 of these around a core. Each one gets a partner, which is like a sticky tail. And so you've created these hairballs with sticky tails. And so by turning on the blue light, this joins, here comes one tail, here comes another tail, you get these sticky hairballs, and those sticky hairballs are sticky enough that they will then phase separate. So I'm going to go back to the talk. I'll try to answer some more questions in a while. Okay, so I was just mentioning that these systems are rather complicated, and we're theorists, and we like really simple systems, because once it gets too complicated, there are too many moving parts for us to model in a way that's interpretable. And so there's a kind of continuum here. Again, I've borrowed this from a nice article by Cliff Brangwyn, reviewing lots of different systems. And over on the left, this is something that comes out of the frog oocyte. If you put a lot of eggs together, there are all of these different droplets. Actually, some of them are nucleoli, some of them are other droplets that come out. And while fascinating, there's thousands of actual different components here, very hard to get a handle on theoretically. This case in the middle is somewhat simpler. There's actually one single protein that when it's expressed and purified and added at high enough concentration to the right solvent will phase separate. But this is already a little bit complicated because this protein is what's called an intrinsically disordered protein. It doesn't fold up like many of the proteins in your body, like hemoglobin, into some specific shape. It's actually just a kind of wiggly polymer, and it interacts with other wiggly polymers through lots and lots and lots of interactions. Some of them are charged, some of them are hydrophobic, some of them are complicated combinations of the two. And so even that is complicated for us as theorists. So the system at the right is something even simpler. And in this case, it's a system of two different polymers that have been engineered so that they phase separate. And each of these polymers is just a series of identical domains. So one of the polymers is sort of a series of identical domains that look like my right hand. The other polymer is a series of identical domains that look like my left hand. And the way these phase separate is one of the right hands gets together with one of the left hands, and this happens multiple times on this polymer, maybe with the same partner or with different partners, left hands coming from different polymers, and that's what causes phase separation. And so I'm going to now spend a few minutes talking about a particular system of this kind engineered from two different domains. And so this was done by Mike Rosen and his colleagues. And what they decided to look at was a system consisting of SUMO domains, which is long name, small ubiquitin-like modifier. Just believe that this is something that real cells express, including the cells in your body. And SUMO folds up, it's basically kind of like my right hand here, and it interacts with a SIM domain, which is a peptide that fits nicely into that cleft, into the SUMO. And so the way that this polymers work is that this binds to that. And so what Rosen and his group have done is that they've engineered a string of these SUMOs. Actually, I'll show you cases just like this, where there are 10 SUMO domains in a row, held together by very, very flexible linkers. Again, these are all proteins. And also these polysims, which are these peptides that interact, these peptides that interact with the SUMOs, and it's a string of 10 of those. And so what happens if you then mix these together? And so what happens is that they phase separate. Now I'm only showing you regions in concentration space right now where they all phase separate. And you can tell the colors just tell you how much of the polysumos there are, how much of the polysims. When there's more overall polysumos, the droplets are rich in polysumos, fewer polysims, and vice versa. But if you go down to lower concentrations, you'll see that this has got a phase diagram where if you don't have enough of the polymers, they won't phase separate. So it's just like the oil and water system. If you only have a little bit of the polysumos and a little bit of the polysims, entropy wins, and somehow they're just dissolved in the water and they don't phase separate. But as you increase the total amount of these polymers, eventually you reach some critical concentration, at which point all of the additional polymer that you put in will go into these phase separated droplets. So we'd like to understand that, but it's already a little bit complicated because there are two components. And so I want to very briefly review something that some of you may have heard about this morning. How do we understand the tendency to phase separate from the point of view of very simple biophysical theory? And so that's what's coming up. We're going to do Polymer Phase Separation 101. And I hope that some of you heard about Flory-Huggins theory this morning, but it doesn't really matter. This is just the simplest equation trying to describe what happens when a single polymer is added to a single solvent. So now we can think about oil molecules being added to water. And the first two terms here, this is the number density of the polymers. This is the log of the volume fraction of the polymers. This is the number density of solvent molecules like water. This is the log of the volume fraction. If you just have these two terms, that comes from the entropy of mixing. That just tells you how much entropy and therefore how much free energy is there when I just mix these systems together. And so if this last term is very small or zero, you get a free energy as a function of the polymer concentration, the polymer, sorry, I got that wrong. My notation was wrong there. Phi 2 is going to be the polymer concentration. And this is a uniform system. If I just put in polymer at uniform concentration, and it goes from 0% of the volume to 100% of the volume, the free energy has this simple minimum like that. And that means that nothing interesting happens. That is, if I give you a particular concentration of the polymer like here, the minimum free energy the system can have at that total concentration is right there for the well-mixed system. So that just says no phase separation. But now there's this other term, and this other term comes from the interactions between the polymer and the solvent compared to the interactions between the solvent and the solvent, like water interacting with water or polymer interacting with polymer. And if the polymer-polymer interactions are more favorable and the water-water are more favorable than the polymer-water interactions, this system will have the tendency to phase separate. And that's reflected in the large value of this parameter chi. The polymers really want to get away from the water. Chi will be large. And then if you add this term, the free energy as a function of the polymer volume fraction will now look like this. And so now something more interesting happens. If I give you a system that has this much polymer added in total, this would be the free energy if the polymer was uniformly dissolved inside the solvent. But what I can see is that if instead I broke the system up into a region which would have a low density of polymer and another region which would have a high density of polymer, then the system could lower its free energy. And the way this is done is you can look at whether the free energy will be lowered if there is a tie line or a tangent line that lies below the free energy curve. And in fact, here, of course, you see that anywhere for concentrations below, above this point or below this one, and through this whole region, it will be favorable for the system to break up, not be one single homogeneous phase, but break up into two phases, a dilute phase and a dense phase. And so that's phase separation. If I added those polymers to the solvent, the free energy would be minimized if the polymers condensed into a dense phase at this concentration, and the rest of the system had a low density of polymers, mostly solvent, at this concentration. And so again, going back to our oil and water, I imagine I add a little bit of oil to water, I'm down here. This is still the free energy minimum in that regime. Oil is just dissolved in water. I keep going, adding oil. Finally, I reach this point. Aha, now any additional oil that I put in will not be at this concentration. It will form droplets at that concentration in a background at this lower density. And if I keep adding oil, the same thing happens at the other end. Eventually, I get to here where I'm back to one phase again. Now, in this case, the system is mostly oil with little droplets of water. And finally, it's just a single phase, which is mostly oil with a little bit of water dissolved. So I would be remiss at this point if I didn't tell you one more thing about this, which is whether this will happen spontaneously or not. That is, if I suddenly mix this oil in water, will it phase separate, or will it have to wait around a while for droplets of oil to begin to form? And so that's kind of easy to understand. Let's look at this diagram on the left. And the idea is, if you notice this, even in this region between this concentration, this concentration and that one, the curve of the free energy has different curvature. So in this region, the curvature is downward. That means that if I give you a concentration right here, anytime I break the system up into a lower density region and a higher density region, I could imagine drawing one of these tie lines. That point will, on the tie line, will be lower in free energy than the point up here. And that means that the system can lower its free energy even by making a little bit here a little denser with polymer, here a little less dense. And so it'll spontaneously phase separate as soon as the system appears. But here there's something else going on. In this region, I'm in the in what is the two-phase region. So the true ground state is phase separated, but the local curvature is upward. That means that the system can't phase separate by making a region of a little higher polymer density or a little lower polymer density. It really needs to know about this state over here of very dense polymers. And so in this region, the ground state is phase separated, but it won't form spontaneously. The system has to wait around and fluctuate until by chance the polymers happen to get together in a little dense region. And then the system will realize, hey, this is a great state. Other polymers can join it, and these droplets will. Now, biology rarely relies on chance. And in fact, in biological systems, often this kind of phase separation is nucleated. So there'll be little particles in the system that are just waiting there for the right conditions, and they can form the centers of these condensates. So the system doesn't have to wait around for random events. So if you put all of this together, then ask, what does it look like as a function of polymer concentration and interaction strength? For weak interactions, this region just says it's one phase, just going to be dominated by entropy. They're just going to be polymers dissolved in solvent. But as you go up in interaction strength, you enter this region here, which is the two-phase region. And if you first come in, this is the region where the ground state is phase separated, but it doesn't happen spontaneously. And then there's this thing called a spinodal line. And if you're inside that region, the phase separation happens spontaneously. Okay, so now you've got everything you need to know about phase separation to understand what we try to understand this system of SIM and SUMO polymers. And what we did is really very, very simple. We did some simulations that I'm going to show you. And in fact, they're so simple, it's almost embarrassing. So, but let me actually pause a moment, see if there are any questions on what's just come before. Okay, I have a question. How does the cytoskeleton affect phase separation in living cells? Here's the answer. It just keeps everything in place, or does it play more roles? I guess it plays two roles. One is it definitely keeps things in place. If you have a cytoskeleton of rigid polymers, imagine one of those droplets forming, it actually could get stuck inside of the mesh here, can't actually move or grow anymore. That's one aspect. But it's also true that in some cases, the cytoskeleton can be a site of nucleation. So just as I mentioned that there are nucleators, the cytoskeleton itself can be a nucleator, or nucleators can bind to the cytoskeleton. So that can be the place where phase separation happens. So I'm going to ask, what can you infer about the droplet phase separation from its constituents, protein structure, etc.? This is complicated. You can tell by bioinformatics, typically whether proteins are intrinsically disordered. So that's a clue that they might be involved in phase separation. But more generally, it's hard to know by looking at a protein. Whether it has sticky parts for other proteins is less clear. It is true, though, that systems with many sticky domains typically are the ones that are strongly phase separated. So if you see many repeats of the same domain, that's also a clue that the system might be one that phase separates. And so, okay, could you please speak to how liquid phase separation works under the paradigm that a cell is in disequilibrium? It seems to me that phase separation is moving towards an equilibrium state. Yeah, it's a good question. The cell is in overall disequilibrium. There's energy is being used, proteins are being made, DNA is being transcribed, and so on. But typically, the timescales for phase separation are such that locally, the system has time to reach equilibrium, even though globally, things are still changing in a non-equilibrium way. So non-equilibrium may be present in, say, modifying proteins by adding phosphate groups to them, but then those proteins will phase separate spontaneously to reach an equilibrium state. I think I'd better move on. I'll try to come back to some of these questions next time. Okay, so coming back to our very, very embarrassingly simple simulations, what we did was we modeled the SIM-SUMO system where there's 10 SUMO domains on one polymer and 10 SIM domains on another, using a two-dimensional lattice. So the idea is that this is a square lattice. Every site here could be occupied, say, by one of the SUMOs or one of the SIMs or by nothing. And we also allow the possibility for a SUMO and a SIM to live on the same site. But we don't let the SUMOs overlap or the SIMs overlap. And so if you put these things down, just let them move around, this is what you get. It doesn't look like phase separation, but that's okay because at this point there's no interaction. So this is really just like a layer of these polysumos on top of another layer of these polysims, and they don't know about each other. So then what do? Well, we want to see what happens if we let them interact. And so we can turn on the specific interactions, giving them strong interactions. So now when a SIM and a SUMO are on the same site, there's a strong favorable interaction. And now we do what are called Monte Carlo simulations, where we just propose moves. Oh, try moving this polymer there. Try shifting this group of polymers over there. Calculate the new energy of the system. And if that's lower, we accept the move. And if it's higher, we accept it with some small probability depending on the temperature. And so let's see what happens when we turn on those interactions and do these Monte Carlo simulations. Well, something happened. Clearly, the polymers know about each other now. And you look at this and you say, no, I don't know. Is that phase separated or not? Well, let's take a close look. So over in this region here, you'll see a kind of dense region. And this is filled with both polysumos and polysims. And each polysumo is interacting with several polysims. Each polysim is interacting with several polysumos. It's kind of a tangle of these snakes. But there's something else that can go on, which is over here, for example, there's a polysumo and a polysim, but they're not tangled up. They're just doing exactly what the other one is doing. So they formed a kind of partnership or a dimer pair where all of the sites on the polysumo are bound to all of the sites on the sim, and they formed a very happy dimer that has all of these sites satisfied. So it is a kind of phase separation between these dense phase of entangled snakes and this dilute phase. But the dilute phase is not individual polymers, it's polymers that have gotten together with favorable partners. Now you're saying, yeah, I don't know, I kind of like those droplets. Those look more phase separated to me. Well, in fact, those droplets also have some weak non-specific interactions. In addition to the SUMO binding to the SIM directly, they tend to interact through some hydrophobic interactions. And so we can add that kind of small additional interaction between neighboring polymers at a very low level, just 0.2 thermal energies, and see what happens. And then we get something that looks more like phase separation, pretty much as much like phase separation as you're going to get in a system that's a two-dimensional square lattice. Now, it's good to trust me, but you should always be skeptical. Maybe this energy is actually enough to have made the system phase separate. Well, we can check that. We can turn off the strong interactions and see what happens. And in fact, if we do that, the system breaks up. So I did want to point out something to you. So let me go back for just a moment and think about what's making this system phase separate. Let's go all the way back to here. And so we're used to the Flory-Huggins theory, where I argued, well, entropy tends to make things want to dissolve and be one phase. Energy makes them want to get together and phase separate. But if I look at this system, I notice that actually all of these strong bonds are formed. In the dense phase, they're formed with many different polymers. In the dilute phase, they're formed with one other polymer. But actually, all the bonds are formed. So this body here and this here have the same energy per polymer. So what's going on? If we don't have an energy difference, what's causing this thing to phase separate? And the answer is it's all about the entropy. So in this region over here, the polymers have a lot of conformational entropy, that each polymer can wiggle in its own way, giving it a lot of entropy that way, configurationally, conformationally. But they don't have a lot of translational entropy. They're really stuck right where they are inside of this big cluster. By contrast, this little dimer here, it can move around as much as it wants. It has a lot of translational entropy, but it actually doesn't have that much conformational entropy, because what's happened is when I tell you what one of the polymers is doing, the other one has to do essentially exactly the same thing. So basically, they've given up half of their conformational entropy. So now this phase separation is a competition between a state which has lots of conformational entropy but low translational entropy, and another state that has lots of translational entropy but much less conformational entropy. And that's what's driving phase separation in this system. So let's move on to, I want to keep going. This simple simulation led us to think about, well, is there a theory we could write down, like the Flory-Huggins theory, that would take into account this very important fact that these domains, the SUMO domain and the SIM domain, interact in a one-to-one fashion? That is, once a SUMO has bound to a SIM, it can't bind to two SIMs, and the SIM can't bind to two SUMOs. And that's different than the case, say, of Flory-Huggins or oil and water, where once you have an oil molecule, it can join another oil molecule on its right, another one on its left, another one in front, another one behind, and so on. So these one-to-one strong interactions are something different. And this had been addressed previously in some beautiful work by some of the leaders in the field of polymer physics, Semenoff and Rubinstein. They thought about solutions of stickers, where polymers would have these sticky sites that would stick to each other. And they were largely considering the case where there was only one type of sticker, whereas we're talking about the case where there's two. There's SUMOs and there's SIMs. And so we have to consider what happens when you have those two different types, and actually some interesting things happen. And so I won't go into it, but we can write down a free energy density just the way that Semenoff and Rubinstein did, or that Flory and Huggins did, now including the fact that there are these two different types of domains on these polymers. One polymer has lots of SUMO domains, one has lots of SIM domains. And I want to thank my student Bin and Jun Zhang, who you heard from this morning, who were responsible for doing the hard work, not only to doing the simulations, but also deriving this formula. So what do we do with this formula? It gives you a free energy as a function of two concentrations, concentration of the SUMOs and the SIMs, and what you get is a landscape of free energies here. And the blue is low, the red is high, and what you can't see here is that these two blue domains here, they're basically like what you saw for Flory-Huggins. They are basically these two local minima, and as a result, there's a tie line, a tangent line, that stretches between these two minima that actually lies below the free energy that you would get if you had a well-mixed system of polymers of SIMs and SUMO. And as a result, this system separates into a dilute phase and a dense phase if the concentration is in this regime. And we can look at this from the full point of view and look at the full phase diagram you get now, not just in the space of concentration of one polymer, but of the concentration of two polymers dissolved in a solvent. And so just like in the experiments on SIMs and SUMOs, where we saw this two-dimensional phase diagram, June is able to derive a two-dimensional phase diagram using the theory. And you can see that there's over here, if I have lots of SUMOs and no SIMs, the system doesn't phase separate, vice versa over here. But if I have an intermediate number of SIMs and SUMOs and enough of them, then the system will phase separate. And these are the tie lines. That is, if I give you a point on this line, what will happen is the system will break up into a dilute phase at this end of the line with a little bit of SIM and a little bit of SUMO. And these will typically be dimers. They will not be individuals. They will be stuck together, but there will still be a low density of these dimers in the solvent. And at the other end of this line, there will be this tangle of snakes where polysims and polysumos have gotten together and interacted with a lot of their partners in this high density phase. All right. So now we see that we can understand in terms of this, simple ideas of phase separation, how it applies in this case where I have two different polymers. So what we've learned is that you can have phase separation driven by entropy in this system where you have these strong one-to-one interactions, case of translational versus configurational entropy. And something to keep in mind for the rest of the talk is that the low density phase can be dominated by things other than free polymers. In this case, it's dominated by these dimers. So I want to use the rest of the time to talk about an actual example in nature of one of these systems composed of these two different types of polymers that only phase separate when they interact with each other. So let me start though by having a question and answer break. See what questions have come up. Okay, in principle, the droplets form with a spherical shape to minimize surface area and therefore minimize the energy. That's good. If one were to confine the mixture in a small box in such a way that the sphere were not the minimum shape, would the droplets form other shapes and so on? So it's certainly absolutely the case that if you constrain these droplets in some way, for example, if they're embedded in one of these actin networks, the polymers will start to grow as spheres, and then they'll run into these filaments, and they'll sort of say, well, I'd like to keep growing, but I can't grow through that filament. So they'll form these blobby bulbous shapes. And in fact, if you look inside of cells at the nucleoli, you'll often find that they're sort of spheres, but they're perturbed. And, and it's clear that they're trying to form a low free energy state in a confined region. So that's absolutely correct. Would dimensionality not have an effect on the equilibrium behavior? Real systems is in 3D. How can you know the 2D results correspond to 3D behavior? Good, you're taking my advice to be skeptical. I will show you some simulations in 3D near the end of the talk. Definitely dimension does have an effect, but the principles of phase separation are essentially the same in 2D and 3D. You can write down the same kind of free energies, looking for cases where the system lowers its free energy by breaking into two phases. And of course, there's also the surface tension. It's a line tension in 2D. It's a full surface tension in 3D. Good question. Can molecular dynamics simulation or a combination of Metropolis, Monte Carlo, and molecular dynamics be used for better results? Well, it depends what you want to do. Molecular dynamics is very good if you're interested in the dynamics of a system, how it actually moves around, or if you're interested in a system where there are continuous movements. So, for example, we talked about lattice simulations. The way polymers move around in a lattice is really weird. They can't just kind of unfold. They have to make these abrupt movements. So if you want to study the kind of smooth movements that real polymers make, you might want to do molecular dynamics in a continuous system. But if you're just interested in thermodynamic questions, like what are the phases, then doing things on a lattice, doing things with Monte Carlo are just fine. So I'm going to move back to my talk for a little while. Okay, so now it's time for the long-awaited Pyrenoid. And so again, why do we care about the Pyrenoid? Well, we'd be in trouble without the Pyrenoid, because as I mentioned, it's responsible for fixing CO2 from the atmosphere. And most of this is happening in these microorganisms called algae, but some plants also have Pyrenoids that fix CO2 out of the air or dissolved in ocean water, and use the energy from sunlight to turn this into organic carbon, making sugars, proteins, and so on that go up the food chain. So here's some beautiful images of the kind of algae that do this. We're going to focus on this one, kind of an ugly guy, but really interesting, called Chlamydomonas. And it is the favorite organism of my colleague at Princeton, Martin Jonicus. Now, Martin is interested in Pyrenoids for many reasons, but one of which is that Pyrenoids are really good at their job. By concentrating CO2 inside of this little organelle, they become more efficient at using the light from sunlight to make organic carbon. And it turns out that many of the plants that we rely on, like wheat, are not as clever. So they don't concentrate CO2 in this way, and they're less efficient doing photosynthesis. And so Martin has a dream to actually understand the Pyrenoid well enough that he could engineer it into food crops like wheat to make them more efficient. And we're talking about like 50% more efficient or more. So if we're going to need to feed a lot of mouths on the earth, this is a really great goal. In the meantime, there's lots of interesting cell biology that we have to understand in order to make this dream a reality. And so we've been working with Martin to try to understand the biophysics of the Pyrenoid. So let me tell you a little bit about the Pyrenoid. So here is an electron micrograph of one of these chlamydomonas or clammy cells. It's an algae. Of course, it's got lots of things in it, a nucleus, lots of other things going on. It's also got a chloroplast, which is false colored green. This is where the photosynthesis happens. The energy of sunlight is harvested to do work, including capturing CO2. And so here is where that happens. This is the Pyrenoid. It's surrounded by these blobby bodies, which are starch, which is a way of storing the energy in organic carbon. And inside of the Pyrenoid is this enzyme called Rubisco. And Rubisco is great. It uses energy from light to take CO2 and strip off the carbon and combine it to make eventually sugars. And so Rubisco is beautifully evolved to do this. It looks, here it is. It's got this more or less 8-fold symmetry, and everything is great about Rubisco except one thing, which is that it's really slow. So it's only doing a few reactions a second. And so in order to actually capture enough CO2 to make this cell grow, it needs tons and tons of Rubisco. So this whole organelle is basically as jam-packed as it can be with Rubisco. And so you can ask, well, okay, why is it in there? Is there a membrane around there that's holding it in, or does this starch actually form a complete barrier? And the answer is no. There's no membrane, and there are big holes in the starch. So how come the Rubisco is staying in there? That doesn't seem to make sense. And so Martin solved this problem in some of his work, and he found by doing genetic engineering on this organism that there's another important component inside of the Pyrenoid. And it's one of these intrinsically disordered proteins. It does not fold up into any particular shape by itself. It was given by Martin the modest name of EPIC1. And so EPIC1 has several repeats of sequences. And these 4 repeats are a clue that it might be involved in phase separation. As I mentioned, it's potentially multivalent, right? It's got the same binding site several times, just like there was the same SUMOs or the same SIMs in the engineered system. And in fact, it really is required for phase separation with Rubisco. And here's just an image showing that they co-localize together inside of the Pyrenoid. Here's the EPIC1 and here's the Rubisco. But one can actually do experiments to show that EPIC1 by itself does not phase separate. Rubisco by itself does not phase separate. But you put them together and they will phase separate. So that's great. It's one of an example of this 2-component system, but there's actually a little bit more to it than that. So, so first, it is liquid, and this is tested in living cells. You can't just test it by sort of squishing them because there's too many other things in there, but you can label them fluorescently, and then using intense laser light, you can bleach some region so that it stops being fluorescent, and then ask, does it stay like this, as if it was a solid, and this part would be bright and that part would be dark? Or is it like a liquid where the bright molecules eventually diffuse into this region and the dark ones diffuse out, and it comes to become a single uniform brightness? And here's a series of images showing that, in fact, after this bleach, boom, it recovers in a period of a couple of minutes. This turns out to be important because there are components in this Pyrenoid other than the Rubisco and the EPIC that have to move around. In fact, there's a particular component that has to rescue Rubiscos that have gotten stuck because they've bound to oxygens instead of to CO2. I'll show you the movie. It's very quick. The bleach happens right at the beginning, so get ready. Bleach in the upper corner. Go. It's bleached. And then over a period of 2 minutes, the fluorescence recovers, showing that this is liquid. Okay, so 2 components, phase separated, liquid. Are we done? Still not done. So what else do we see about these Pyrenoids? Well, something really interesting happens as these cells are going through cell division. So here's an image just of the Pyrenoid through imaging the Rubisco, the rest of the cell is here, you can't see it, and it's condensed. This is actually a Pyrenoid. But just before the cell goes through division, something interesting happens. A lot of the Rubisco and a lot of the EPIC1 leaves the Pyrenoid, goes into the rest of the chloroplast, and then after cell division, these condense again into two daughter Pyrenoids. So is this an accident? Nope, it happens every time. Here's the same cell going through another couple of divisions. It's a live cell, it's actually rotated. So these two Pyrenoids again partly dissolve, and then after it's divided into four, it's already now condensed. And so there's lots of questions here. There's how questions and there's why questions. The why question is maybe a little bit answerable here. It turns out that sometimes when the cell divides, instead of the body of the Pyrenoid getting divided in half and going half to each daughter, it actually all goes to one daughter. The division kind of squirts the Pyrenoid out to one side. And so in that case, even the unlucky daughter has a lot of the building blocks, the Rubisco and EPIC, to form a new Pyrenoid. So maybe that's one reason that why this happens. But then there's the question of how. We've just been arguing that phase separation is physics. Physics didn't change in the few minutes between the cell here and the cell going through cell division. So what is it that could have happened? So let's show this as a movie first. This is sped up. You see that this kind of weird spreading out of the material, going back to a one-phase system, then back to phase condensed in a system, back to one system, condensed again. So how do we understand that? So this is happening fast. It can't be that there's new protein being made or old protein being degraded. It has to be the same protein that wants to be condensed, then it doesn't, then it does again. Well, this can happen through chemical modification, but how are you going to modify all of those proteins? That's hard, or at least it's hard to modify them a lot. Here's where maybe the magic numbers come in. So magic numbers say that you don't necessarily have to modify things a lot in order to get a big change in their phase properties. So let's take a look at what could cause this rapid reorganization of the Rubisco. And to do that, we're going to go back to our very, very simple lattice model simulations, back to these 2D square lattices. Instead of PolySim and PolySUMO, though, we've got Rubisco and EPIC1. And Rubiscos are going to be little bricks, 4x2, that's 8 binding sites, like the 8-fold symmetry of the Rubisco. And the EPICs will have a certain number of binding sites. And so we'll consider first maybe that there's maybe only 3, maybe there are 5. I've left a blank here for 4. What do we see if we mix together these little polymers with these bricks with the assumption that they like to stick together? And what you see is you get some kind of phase separation. They tend to form these clusters. And if I use the same number of monomers, but I string them together in longer polymers, that tends to favor phase separation. These longer guys can connect to more of the Rubiscos, and indeed I get kind of larger, more condensed clusters. So what would happen if I just give you an intermediate length, L equals 4? Is it just going to be right in the middle? Well, that would be boring. So I see that if I have just changed the length of the EPIC1s to 4, something very different happens. I don't get such a phase condensed system. And if I look closely, I can see why. Because in addition to the possibility of having a condensed phase like this, there's something else that can happen. And that is that one Rubisco with 8 binding sites can get together with 2 EPIC1s, each with 4 binding sites, and they can form a happy family, where all the binding sites on the Rubisco are bound to EPIC1, and all the binding sites on the EPIC1s are bound to Rubisco. So they form these little trimers, much like the dimers we saw of the PolySims and the PolySUMOs. And so these guys, they have a low energy state, all the binding is done, and they have lots of translational entropy. They can move around as much as they want. And therefore, that favors this single phase state. And so if you look down here and ask, when do you get phase separation as you increase the total amount of the Rubisco and EPIC1, going to the right at strong interaction strengths, here's where you start to see phase separation if you have length 3. With length 5, indeed, it happens earlier at lower concentrations. The system likes to phase separate. But with L equals 4, It doesn't like to phase separate until really high concentrations when these are so dense that they don't have much room to move around, so the translational entropy isn't so important, and they can get more conformational entropy by forming structures like this. So we're getting near the end of our time. I have a few things that I want to say very quickly to reassure some people in the audience. Let's just say that this could, in principle, be a biological magic number effect. So let me quickly go forward, and I'll come back and have a question and answer break at the very end. So for those of you who read the paper, you noticed that we made a case that having flexible polymers and bricks is different. If you look at the case where, for example, if Rubisco wasn't rigid, but it was a flexible polymer, you would see a magic number effect. That is, when I have, for example, epic ones now not of length 4, but of length 8, another magic number, I could get a very suppressed onset. The average cluster size is really small in this case, showing you that at this concentration, if I have an equal amount of the epic one and the Rubisco, the system doesn't want to phase separate. But as soon as I get to rather imbalanced stoichiometry, more Rubiscos than epic ones or vice versa, kind of relaxes to a state where the system phase separates. And this is quite different, though, if the Rubisco is a brick, right? So what's different? I've just taken this Rubisco, and instead of having a floppy polymer, I've made it a rigid brick. And so the difference has to do with how much entropy the polymers give up when they get together compared to how much entropy a polymer and a brick give up. These polymers, when they were by themselves, could have done anything. Then they form this polymer pair. They've actually given up a lot of entropy, conformational entropy, to do that. So they might actually prefer to be in this condensate. However, when I have a polymer and a brick, the polymer can do various things inside the brick. This is not the only conformation. And the brick didn't have any conformational entropy to begin with, so it has nothing to give up. And that's why these two systems are different. It depends on how much entropy the components have to give up to form these dimers compared to what they have to do when how much entropy they give up when they join the condensate. For those of you, again, who are interested in higher dimension, June has done some beautiful simulations off lattice in 3D. This looks like your SIM-SUMO poly-SIM system. And you can see that when you let this system evolve now under molecular dynamics, it will form a condensed phase and a dilute phase. This difference here has to do with the different stoichiometries. But to be reassuring to you, there really is still a magic number effect. So if you look at the phase boundaries, when the interactions are not very strong, this is the dilute phase boundary telling you how likely the system is to phase separate when I have long polymers of length 14 and some other length. And it doesn't seem to really matter whether the other polymers are length 13, 14, 15. It's just that the longer the polymers are, the easier they are to phase separate. But then as you increase the interaction strength or equivalently lower the temperature, you see this thing happen where 14 is special. And this is the magic number effect. It says that, yeah, this system loves to phase separate when it's length 14 and length 12 or length 14 and length 13 because they can't form a happy pair. But when they're length 14 and 14, they can get together and be perfectly energetically satisfied. And that suppresses the phase separation. So you have to go up to much higher density in order to phase separate. And of course, as you noted in the epic Rubisco system, it can also be a magic number if one length 14 gets together with two length 7s. So again, another magic number effect. Okay, so what, if anything, does this have to do with biology? Well, the idea that we're pursuing is that maybe this dynamics of the Pyrenoid, where the system dissolves the Pyrenoid and then gets it back together, could be based on magic numbers. That is a very small change. Changing the number of binding sites on one polymer just by one could have a big effect. So for example, now imagine just for kicks that the epic ones are length 3 because one of those binding sites has been chemically modified. You could get a phase separated system, kick off that chemical modification till they're length 4, and you get a magic number effect. And all of those trimers of Rubisco and 2 epic ones will dissolve out into the chloroplast. So I come to the end with some conclusions. Just generally, the field of cell biology has been revolutionized by the idea of these non-membrane bound organelles or phase separated condensates. They're everywhere in our cells, and I really mean our cells. For these two-component multivalent systems, which are also common, entropy can drive a phase separation by itself. And an important thing to remember is that even though the condensed phase looks interesting because of all of this tangle of different polymers, the dilute phase is also important, and the small oligomers there can be very essential to determine where the phase boundaries are. We're interested in the possibility that this magic number effect may influence phase separation as well as its dynamics, which I didn't have a chance to talk about today. I do want to thank the people who did the real work. Yaojin Zhang, my student Ben Weiner, who many of you heard from this morning, former and present students, postdocs, and colleagues, and support from the CPBF and the NSF. And now I will be happy to take the last minute or two to answer some questions. Does the phase separated states in the system of two polymers in a solvent also obey Ostwald ripening? Yes, absolutely. Ostwald ripening is the statement that if I have two droplets, a big one and a small one, the actual free energy of the system would be lower if they were together. Now, one way to get that is to have them merge, but the other way is that the small droplet will have a higher density of the dilute phase around it than the big one. So there'll be a net flow of these dilute phase material from the small droplet to the big one. The small one will eventually shrink, disappear, the big one will grow. That's Ostwald ripening. And that will definitely happen in these more complex systems. What will have to move between the small polymer and the big one would be, for example, dimers of SIM-SUMO or trimers of Rubisco and EPIC 1s. Yes, what could change the length of EPIC 1 biologically? We're imagining a chemical modification. Biology is filled with enzymes that add phosphate groups or acetyl groups or methyl groups or remove them. And so that's what we're imagining happening. And it is known that EPIC 1 does get phosphorylated. We're still, I should say, there's some questions here about what's our current status. We're definitely working. We've been able to reconstitute the EPIC Rubisco system in the test tube. My student Guanhua He, working with Martin Jonicus, has done that. And so we're moving on to look at what happens when we change the number of binding sites on EPIC 1. Yes, and someone's asking, oh, that's moved on. Is there any application of this model except just theoretical knowledge? Yeah, absolutely. Oh, good question. Can it be applied to speed up sugar production, for example? That is certainly the hope. Well, I mean, the Pyrenoid in the algae has certainly evolved to be very good at its job, but as we try to get these systems to work in crops like wheat or corn, understanding the system and possibly making it better is on the table. But I should also say there are people very interested, for example, in making biofuels using cells. And one of the big ideas is to get some of those reactions to happen inside of these condensed regions. And so being able to make those form when you want that reaction to happen and dissolve when you don't is a big question. And being able to use the magic number effect is a real application. And one last question is, okay, any relation to chaos in the phase separation processes? That I don't think we see chaos, but we see a lot of disorder. That is, every time you do a simulation on these systems, droplets will form in different places, diffuse around in different ways, and merge. But on average, the behavior is not chaotic. It's more sort of, it's more random, but predictable. And with that, I will sign off and thank you all for joining us today. Take care.